I'm a postdoc here at the Smithsonian National Zoo, and I work here at the Genetics Lab. Working at the Smithsonian has been a dream of mine for a long time, and it's really incredible to be here surrounded by a community of amazing scientists. As a conservation biologist, one of the things I'm most interested in is what happens to animals when they are confronted with a deadly disease they have never been exposed to before. When humans travel to new places around the world for business and leisure, we unknowingly bring with us insects like mosquitoes and fleas. And then, in turn, those insects can transmit diseases to animals that have never been exposed before, and so have had no opportunity to evolve resistance. These animals are extremely susceptible to the new diseases. Let's take a look at a species that serves as a really powerful example of what can happen when a deadly disease is introduced. Prairie dogs are a type of ground squirrel that lives in huge colonies consisting of family groups. They have a complex communication system for warning each other about different types of predators. They have different alarm calls for hawks, coyotes, snakes, and people. Prairie dogs are really important for grasslands because they're a primary food source for dozens of animals, and when they build their burrows, they aerate the soil and bring nutrients to the surface where they are available to plants. Historically, they were an emblem of western grasslands, sharing the prairies with bison. But in the early 1900s, bubonic plague was introduced to North America by accident. And yes, this is the same plague that wiped out a third of the population of Europe in the 1300s. Unfortunately, prairie dogs are even more susceptible to plague than humans are, and their populations have been reduced by 98%. Obviously, this has been a really devastating disease for prairie dogs. In the last few years, people have started to notice that a very small number of prairie dogs, about 1%, will survive a plague epidemic. That led me to wonder if we could figure out the genes that are responsible for surviving plague. This is one of my main research goals, but it's not a straightforward problem to tackle. Because prairie dogs are not closely related to humans, and they're not a model system like mice or flies, we know essentially nothing about the genes that control disease response. As a result, my work was previously limited to non-coding DNA, which actually tells us a lot about things like migration and inbreeding, but it doesn't tell us a lot of the things we really want to know, like how some individuals can be resistant to this really deadly disease. Fortunately, genetics has entered a new era. With the invention of next-generation sequencing technologies, biologists working on animals in nature have all these tools available to us that were out of reach before. We can now use very tiny amounts of animal tissue or blood to sequence an entire genome. For prairie dogs, I use a little ear punch, it's just like getting your ear pierced, to get the DNA that I need. Then I extract DNA from this tissue, break it into small, overlapping pieces, and then I prepare those pieces to be sequenced. After I get the sequences back, I have to do a lot of programming to sort through the data and fit all the overlapping DNA sequences together. Only after I assemble the prairie dog genome can I figure out which genes confer resistance to plague. Programming is something I never expected to enjoy, but I've been pleasantly surprised to find that I love it. It's not easy, but the challenge is part of what makes it so fun. Sequencing a genome is a critical first step in understanding susceptibility to disease. Not only this, but the availability of a genome will also help other scientists who are studying various facets of prairie dog biology, including their complex communication system and social behavior. If you would like to help, please consider donating to my campaign and share this video with your friends and family. Your contribution will help cover my lab supplies and the sequencing costs for building the first prairie dog genome. Thank you for supporting my career as a female scientist.